there. So I do apologise for not being able to do this in the live session, but as promised, here is a recording of case 4 because I know you seem very interested in that. So I have a 25-year-old female. They have been involved in an RTC. They've grouped as OPOS. They don't have any historical antibodies. You're aware they've had a previous pregnancy, and 4 units of red cells have been requested for this patient. But I'm just going to revisit the RH nomenclature table because I'm just sort of wanting you to understand what is happening in terms of the phenotyping and the shorthand that we use. So when I discussed the R1R1 panel previously, what this means is that is a panel which is negative for the little c antigen, because the phenotype for an R1 is D-pos, big C-pos, little E-pos. So that was the sort of makeup of the panel, but I've included the frequencies here. And I just wanted to sort of revisit this just when we're talking in relation to the antibody that we're hopefully going to identify shortly. Just when I'm talking about phenotypes, it just gives you a bit of information to understand when we talk about what the most likely phenotype is, because based on your genotype, you can sort of assume that somebody's phenotype is one thing when genetically it may actually be something else, because we tend to just base it on the most likely one based on the donor phenotype. So this is your shorthand from your panel. So we were talking before previously, we're looking at the R1R1 panel. That's D-pos, big C-pos, little E-pos. But in this case, both genes. So while you might just look at one gene and call it R1, this is an R1R1. So this person has 2 copies of this gene. Same applies here with the R2R2. In the middle there, we've got the little r. This is your D-neg, little C-pos, little E-pos, which is your most likely phenotype for a patient who when you would say they were little r. And for that little C panel that we showed you earlier, the R1R1 panel, the only cell line was line 5, which is an RO cell. So that's it, just for your information. So when we're looking at case 4, so this is your panel. So this is your primary panel that you would do in your hospital laboratory. And during the live session, I would have asked you what antibody you have identified. This is a little bit of a tricky one that I've put in because it does look a little bit like anti-little c, but it isn't. So with this panel, when we go through our exclusions, we can actually rule out every antibody that is listed across the top of your antigram, because you can exclude everything in that panel on your first 3 lines. What we do have is positive reactions from 4 through till 10. We've got uniform reactions in IAT, which are 2+ strength. We also have uniform reactions in enzyme IAT, which is 4+. So even though we've excluded every antibody there, we know that there's something going on. So this is where you just need to use those sort of knowledge and skills of your understanding of your antibodies, and kind of to be guessing here, what you might be thinking is, well, I've got something that's enhanced in enzyme. So that could be an RH antibody, for example. So that's, if I was doing this, that's how I would be starting to think about this. So what would you do next? So you could put another panel up to see if that would help you, but you're going to get identical reactions to this. You could refer to your local reference laboratory if you wanted to see what they thought that this might be. What I would do is I would phenotype the patient. So it's looking like an RH antibody. So the sensible thing to do would be to perform extended RH and K phenotyping. The patient's autocontrol is negative, and as with the previous examples, this phenotyping uses IgM antisera, but just be mindful of the autocontrol when you are doing your phenotyping. And with this patient, the big C-pos, little C-pos, big E-pos, little E-pos, K-neg, and as usual, we've done our reagent control, and that is negative. And this patient, based on the RH shorthand, is an R1R2, and I've put there that that's the most likely phenotype, because it could be that they have a different genotype, but most likely, based on the sort of frequency of these 2 phenotypes, it's most likely that this patient is an R1R2. So you can see there, you've got your positives and you'll have your negative control there. So this antibody is an anti-F. The reason that we know this is we've got to use the understanding of how antibodies sort of behave. So what I have done here is I've put in some F negative and F positive phenotypes. So anti-F can be a little bit complicated to get your head around. So if you look in here, F negative phenotype, you've got R1R1, so that is D pos, big C pos, little e pos. That person has 2 of those genes, so that's why they're an R1R1. But if you look in the positive column, so I have a little r on the first line there, you can see in red that this patient, the D negative, but the little c pos, little e pos on both of the genes. So how anti-F works, it's actually a compound antibody. So what that means is, even though this looks like an anti-little c, it's not an anti-little c. What it is, a compound antibody, if you think of the 2 of them, because when you inherit an Rh gene, they can come across together. Anti-little c, little e compound, i.e. anti-F, is actually, if you think of the 2 genes, it's not little c and little e on one gene. They sort of merge together almost to form this compound antibody. So it is anti-CE or anti-little c, little e, and we'll call that anti-F. So you can see they have put there's some other positive combinations there. So any patient where the gene has the little c and the little e on the same side, that is F positive. So you can see on the R1R1, R2R2, and the R1R2, they are all F negative, because even though the R1R2, for example, has its little e positive and its little c positive, so it has those antigens. What it doesn't have is the little c and the little e on the same gene. Therefore, it is F negative. Hopefully that makes sense to everybody. So one of the things you can do is you can send that across to the reference laboratory. So you can see here, this is the R1R1 panel, which is the same as what we used for the anti-little c. And if you see across that line there, that's the one line that is little c positive when we use that for the R1R1 panel. But actually what you can see on here is that this line is F positive. So it also has the little c and little e. And because this little c and little e are on the same gene, this cell is an F positive cell. So because we've identified an anti-F, because I'm going to show you in a moment how to get your exclusions to show that it's not anti-little c, this cell is where they're in combination with each other, and you've got a positive reaction there. So if we go back to our original panel, if you look more closely, the only positive reactions that you have on this panel are where the little c and the little e are on the same gene. So cells 4 through to 10 are all F positive cells. So if we were to go back to that table, you can see these are all F positive because the little c and little e live together essentially. And that's why you get this positive reaction. So this is why F is a little pesky one, because it kind of looks like a little c, but it's not, because you can actually exclude the little c, because cell 4, if I go here, so this is confirming the anti-F. So you've got 2 positive examples and 2 negative examples of the anti-F. What you do have, if you look very closely, sorry, it's if you look very closely at line 3, should I say, that is the little c positive line, and you can exclude it. But if you notice, cell 3, it's positive for little c, but it's negative for little e, so they don't live in combination on that cell. Therefore, you can show that it's not a little c. Whereas if you had a little c, line 3 would be positive. So that's how you can tell the difference. But the panels don't tell you that this is an F, so it's just having that understanding of your antibodies, how they behave. So it looked like an Rh system antibody because it was positive by IAT and enhanced in enzyme IAT. And it sort of looks a bit like an anti-little c. So that's kind of how we can understand that. But so even though it's not listed, you can kind of see how it behaves, and then it's understanding your phenotyping and this is where your RH shorthand comes in quite helpful as well. So is allo-anti-F clinically significant? So if this was the quiz here, the options are all your RH antibodies are clinically significant. Allo-anti-F is only clinically significant when detected by IAT. Is allo-anti-F clinically significant, or you're not sure? And if you're not sure, that would be absolutely fine. But if we go through, anti-F is actually clinically significant, but this is where you would be checking your BSH guidelines, or things like your antigen fact books would be able to tell you. So it's clinically significant. It has been implicated in HDFN, and if a patient has anti-F, you need to select antigen negative blood. But obviously, this wouldn't be listed on the unit as blood as being F negative, whereas anti-little c, for example, it would say that it was little c negative. So you need to look for a phenotype where the patient is, ideally, you're going to be wanting to select your R1 or your R2. You could technically select R1, but you wouldn't know if genetically that unit was R1, R2. It's very confusing. So your safe bet really is R1 or R2. However, R2 phenotype is quite rare. It's only about 3% of the population, so really you're going to be wanting to think about giving R1s to this patient. And I've said at the bottom there, individuals are F negative when the C and the E are on different genes. So that's how you would know. So what blood would you select for this patient? So I've just given you various combinations there. We've got the R1, the R2, an R1, an RO, or a little r, or you're just unsure. So what I have said there, just to reiterate what I've mentioned previously, you would be wanting to go for your R1, that would be fine because the F negative. Same with R2, same with R1, but with an R1, it would be difficult to know if the C and the E are on different genes. So your safe bet here would be your R1, just because you're more likely to have those available as well. Or if you wanted to, you could discuss with an HSBT who could even perform that cross-match for you. And these are all F negative because the little c and the little e are on different genes. So what your positive ones are, you wouldn't want to be choosing these. So you wouldn't want your RO because that's F positive because the little c and the little e are on the same gene. So you can see there, with R1, it's little e positive, but it's not F positive because it doesn't have the little c. The R2, you've got the little c, but you don't have the little e in combination. They don't have little e. And the R1, you've got both the little c and the little e, but they live on different genes. So I hope that has helped everybody. If you have any further queries, please don't hesitate to contact me. And there are some great resources on the Transfusion Training Hub as well if you want to visit that website. Thanks very much.